Hello students, welcome to the SRBPS virtual classroom program. I am your biology educator Prerna Rathi.
So now let us start today's class. Today we are going to learn about microbes in human welfare, that is chapter 10 of your NCERT textbook. Let us first see what are microbes. Microbes are diverse protozoa, bacteria, fungi, microscopic organisms like viruses, viroids, prions, which are just proteinaceous pathogens in nature. They could be bacteria, fungi, which can be grown on any culture media artificially. Some microbes are disease causing, that is pathogenic in nature. As you can see in this picture, there is a bacteriophage, which is a virus which infects the bacteria. There is an adenovirus and lactobacillus. Now let us see how do we use microbes for the human welfare. Let us first discuss microbes that are used in household products. The first and the most important is the lactic acid bacillus or lactobacillus, the lab. This bacteria is used in our everyday life to create curd from milk. What happens is when the inoculum is added into the warm or lukewarm milk, then as the bacteria starts growing and respiring, it converts the milk proteins into curd. Also, it improves the vitamin B12 level and hence makes the curd healthier than the milk. Then, for preparing dough for breads, batura, etc., we use Saccharomyces cerevisiae, which is known as baker's yeast. Apart from that, in order to prepare cheese, there are different microorganisms that are used. For example, in order to prepare standard Swiss cheese, Propionibacterium shermani, this bacteria strain is used. Now, it produces holes because of the evolution of carbon dioxide. Then, in order to produce Roquefort cheese, Penicillium roqueforti is used. It also gives a very peculiar flavor to the Roquefort cheese. As you can see the diagram of both the cheese, there is Swiss cheese which shows presence of holes, and there is Roquefort cheese which shows presence of this green penicillium. Apart from that, in the southern part of India, there is a traditional drink that is prepared which is called toddy. It is prepared from the sap of the palm trees. It is prepared by fermentation. For this also we use microorganisms. Apart from that, in order to prepare, in order to ferment a fish, bamboo shoots, soya bean and various other food items for preparation of sausages, etc., also microbes are used. Now let us see what is the industrial use of microbes for human beings. The most important once again would remain Saccharomyces cerevisiae, which was used as brewer's yeast, that is to ferment malted cereals and fruit juices in order to produce ethanol or alcohol industrially. Fine. So wine and beer are also produced by the same method, but they are not distilled, whereas other alcoholic beverages are distilled. Next industrial production is the production of antibiotics. Antibiotics, children, are the chemical substances which are produced by some microbes and which can kill or at least retard the growth of certain microbes. The first antibiotic that was discovered was by Alexander Fleming. What he did was he was culturing Staphylococcus bacteria. Fine. And while he was culturing, he saw that there was a mold growing in his culture plate, and around that mold there was no Staphylococcus growing. So he came to know that this mold, that is Penicillium notatum, has or produces something, a chemical that does not allow the growth of the bacteria, and that was penicillin. Ernest Chain and Howard Florey made its full potential and an effective antibiotic. Other products are various organic acids like citric acid produced by Aspergillus niger, then acetic acid, Acetobacter aceti, butyric acid by Clostridium botulicum, lactic acid by Lactobacillus. Fine. There are a few enzymes like lipase. Lipase is a lipid digesting enzyme which is used in laundry detergents and in the detergents which are used to wash our utensils, etc. Then there are pectinase and proteases which are used for clarifying the bottle juices. The third one here is very important. Streptokinase is produced by Streptococcus bacterium and it is used as a clot buster to remove clots, especially during a myocardial infarction, that is a heart attack scenario. Various bioactive molecules are also produced using microbes. The two most important one of those are, first is cyclosporin A, which is used as an immunosuppressive agent, which is used during various organ transplant or grafting surgeries in order to bring down the immuno level of a person or patient to increase the acceptance of the graft or the transplant. Then there is statin, which is produced by Monascus purpureus, which is an yeast, which is a blood cholesterol lowering agent. Now let us discuss sewage treatment. Sewage treatment has been made compulsory by the law of India. The Constitution of India holds it at utmost importance and has started two very important programs for sewage treatment, the Ganga Action Plan and the Yamuna Action Plan. The Ganga Action Plan is now run by a new name called Namami Gange program. The treatment of sewage is done by using heterotrophic microbes which are naturally already present in the sewage. It is done in two steps, the primary treatment and the secondary treatment. The primary treatment is mainly a physical treatment, whereas the biological treatment is done during the secondary treatment of sewage. Let us discuss the primary treatment. As you can see, there is a pre-treatment zone where the primary treatment is done, and then proper sludge digestion in order to produce biogas is done. Let us discuss primary or the physical treatment. It involves first sequential filtration through wire meshes of different size. Then sedimentation, where the grit, small pebbles and soil is removed by storing the waste for long hours in a sedimentation tank. The effluent that is produced here is taken for secondary treatment, whereas the primary sludge that is produced here is used for various purposes like landfills. Fine. So the effluent is very important. Effluent enters the secondary treatment or the biological treatment. The primary effluent is passed into large aeration tanks which have continuous oxygen supply. This allows vigorous growth of aerobic microbes along with the fungi, which forms flocks, so that the surface area for respiration increases. While growing, they consume most of the organic matter and thus reduce the biological oxygen demand. The biological oxygen demand is the amount of oxygen required by per liter of solution to be digested. All the organic matter that is present in 1 liter of solution requires certain amount of oxygen. That oxygen is its biological oxygen demand. Greater the BOD, greater is the polluting potential. So when the BOD reduces, we come to know that it has been now purified. Slowly, as the BOD is reduced, then once the BOD of the sewage water is reduced, the effluent is then taken to the settling tank where the bacterial flocks are sedimented. They are sedimented, they are separated. Little bit of bacterial flocks are kept for activated sludge so that next time inoculum can be made with this. Then this is then sent to the anaerobic sludge digesters, where anaerobic respiration occurs in the presence of certain anaerobic bacteria like Methylotrophus, Methylophilus, many methanogens basically, which lead to the formation of biogas, which is a mixture of methane, hydrogen sulfide and carbon dioxide. The effluent from secondary treatment plant is generally released into the river, because as you have seen, after all this treatment, we have actually removed all the polluting material from it. So as you can see here in this diagram very clearly, first the raw sewage is sent through screens, then from a commutator it is sent to the primary clarifier. After that it goes to the aeration tank, last to the anaerobic digester, and finally the secondary effluent is given into the river.
I hope you all understood what I taught. I suggest you all make complete use of the YouTube channel to enhance your learning. So until next time, goodbye, take care.